we have already seen that one of the things that plants need to have for photosynthesis to take place is chlorophyll. The role of chlorophyll is basically to capture sunlight. Okay, so sunlight energy is captured by chlorophyll. So how can we conduct an experiment to demonstrate that indeed, if this chlorophyll is not there, the process of photosynthesis cannot happen? Okay, how can we do that? But before we look into that, this experiment, what can it tell us? How can we apply it in real life? Okay, so this is where we start with a scenario talking about a farmer who has been growing spinach. Okay, so this farmer notices something. He notices that normally his spinach is actually green and growing well and thriving, but there's some spinach that are having a yellow color, and this yellow color is actually not something usual. That's one. But the second thing that he notices is that those that have yellowish leaves are actually also not growing well. So that's a question that he has. So that means from our experiment that if the plant is lacking chlorophyll, perhaps is having another color, okay, that means that it cannot carry out photosynthesis. And if it cannot carry out photosynthesis, means it will lack the organic compound that it needs for growth. Okay, remember that photosynthesis gives us or gives the plants starch. Okay, starch, and it is that starch that is used for a number of processes, including growth. So if we are having plants that are turning yellowish and they're not having chlorophyll, which is green, then they're not going to grow because of the fact that they're not photosynthesizing well. So that's something that you need to know, and why this experiment becomes very important. It will show us that plants, parts of plants that are not green are not actually carrying out photosynthesis. So, like we have noted, the plants appear green because of chlorophyll. So chlorophyll is a pigment that is used by plants for the process of photosynthesis. What they specifically do, what the plants do with this chlorophyll is that this chlorophyll helps in trapping the light energy. And this light energy is what is used to convert carbon dioxide and water into the glucose, okay, which is later on converted into starch. And then also carbon dioxide is given out. Okay, so in this experiment, we want to determine whether indeed where you have chlorophyll, you will have starch, and that will indicate that there is photosynthetic activity taking place in green parts of the plant. And in this case, we are looking at the leaves. Need for this experiment, you basically need the things that we use for testing of the presence of starch in leaf. So basically, the process is similar. The only unique thing is that you get a variegated leaf. Okay? So what you're going to do on the variegated leaf is basically the same thing that we did when we were testing for presence of starch. Okay? So definitely you need a variegated leaf. You need the tongs, you need containers, you need boiling water, you need scissors, petri dish, you need ethanol. You also need iodine because you have to test for presence of starch using iodine. Okay? So the procedure is as follows. is that, first of all, you first need to get a variegated plant. From this variegated plant, that's when you pluck off or cut off a leaf using the scissor. Okay, so you first deal with the plant, get this plant, expose it to sunlight for 4 to 6 hours. Why do we do that? To ensure that this plant carries out the process of photosynthesis. And when it carries out the process of photosynthesis, and then we test for presence of starch, we shall be able to find it. Okay, we shall be able to find that starch. So the next thing, after that exposure, you cut off one variegated leaf. So that leaf is what we are going to test for the presence of starch in the different parts. The part that contains chlorophyll and the parts that do not contain chlorophyll. So what is the process of testing for starch? The first step is that you get this leaf, boil it for 1 to 2 minutes. Okay, this boiling kills the cells in the leaf such that you don't have any more photosynthetic activity happening or any other chemical reactions happening within the leaf. Okay, it also helps in softening the leaf. The next thing that you do is you transfer this leaf from this boiling water to another container that contains ethanol. Okay, and then put that leaf into that container that contains ethanol, put it in another container or beaker that contains water, and then heat that leaf which is in ethanol until the leaf loses its green color, that is decolorizing. The ethanol helps in that process of decolorizing. Okay, so you heat until the leaf loses its green color. So that's until the leaf, the green color fades and the leaf appears purple. What is unique is that the parts that will have lost the chlorophyll will appear distinct from the parts that do not have chlorophyll. The next step is that you remove that leaf, rinse it with water, okay, and then place it on a flat surface. A petri dish can work. And then after putting it on a petri dish, and then you can get drops of iodine. You can drop iodine onto this leaf. Be keen on noticing where you put in the iodine. You put the iodine on parts which were green, and then the parts which were the parts that were non-green. Okay, and then you notice the color changes. Okay, the parts that will turn out to be blue-black, it means that they will contain chlorophyll, and the parts that do not show any color changes will be those that did not contain chlorophyll at all. So what observations will you make? Initially, the green parts of this leaf and then the non-green parts of this leaf will appear the same. But over time, you will notice that the green parts will turn dark, okay, or blue-black. And then the parts that did not contain the chlorophyll that we are terming as either yellow or white, you will not see any significant change. Okay, and that will indicate to you that the turning to dark or blue-black indicates that there was photosynthetic activity and there was starch in there. And then the parts that remain the same means that there was no chlorophyll and photosynthesis did not take place. So the conclusion is that you actually need chlorophyll for the process of photosynthesis. Why? Because the chlorophyll helps in trapping